It's wonderful to be here. I really appreciate the invitation. I've already had amazing conversations this morning. Looking forward to some more this afternoon. And today what I'm going to talk about, it's a little bit of a detour for me. It's kind of a side project that I've been working on for a while. The core of my research really I do in the Philippines with a long-term study. It's been going for more than 30 years. And we track all kinds of things and look at genetics and public health outcomes and human life history and all sorts of questions. Today I'm going to talk about brain energetics. It really is a bit of a side project for me, but it's something that this particular project was about 7 years in the making. So it took a while to reach fruition. And I'm going to start with the photo of my son because why not? We can do that, right? And he illustrates some things that I think are unique about humans that set us apart and our life history apart. He's about 9 or 10 months of age here, and he's being fed, so he's starting to be weaned, right? He's being fed baby foods, like complementary foods. They're specially prepared, mashed up, cooked, and so on. And he's being fed by somebody other than his mother. In this case, he's being fed by his grandmother. Now, this may seem pretty normal, but it's actually something that is distinct, and it really sets our species apart from most other mammals. What you see here are some basic differences between a chimp and a human life history. So we do wean quite a bit earlier because of these special weaning foods that we have, right? Chimps wean about 4.5 years of age. And we've got a pretty good handle as to why we do that, right? There's a clear reproductive advantage to that. If you wean earlier, then you start to ovulate earlier, you reduce the interbirth interval, and you have a higher fertility rate. And this is one of the reasons, maybe not the most dominant reason, but it's one of the reasons that there are more of us than there are chimps in the world, right? We have much higher fertility. So that piece of the life history story I think we've got a pretty good handle on. But you can see the other piece up here, which is the length of these blue bars, right? Chimps start reproducing much earlier than we do, right? We delay onset of adulthood quite a bit less. And during this period, we slow our growth rate down remarkably, right? And so why we do this, why we slow our growth to the extent that we do, is not something that we have a good handle on. So let's take a quick look at how slow our growth is, just to give you a feel for the magnitude of what we're talking about here. We're going to start with something that many of you are probably familiar with. This is the Kleiber line, showing metabolic scaling of caloric expenditure here on the y-axis. So you can think of this as something like calories expended per day, plotted versus body mass. They're both a log-log plot, and when you plot them like that, you get a nice straight line, right? Now, the main thing to point out here is that the scaling exponent is less than 1. It's 0.75. What this means is that as you go from smaller-bodied animals to larger-bodied animals, of course, overall expenditure is increasing, but it's increasing at a rate that's slower than the increase in body mass. In other words, a kilogram of elephant or a gram of elephant uses far fewer calories than a kilogram of mouse. You get this increase in efficiency as you get to larger bodied animals. Now, there's all kinds of debates as to why this relationship exists, and it has this mathematical regularity to it. But what it tells us is that there's a constraint on energy expenditure. So if you think about energy as kind of a pie of resources that an animal has available to it to allocate across different functions, it's clearly pretty constrained mathematically by something related to body size, right? And not surprisingly, when you look at different components of expenditure, it has the same sort of relationship. So what this shows is growth rate from birth until about 40% of adult size. These are data compiled by Case in a QRB review some years ago, some decades ago. And what you see is the same scaling coefficient, right? It's about 0.75. Given that overall expenditure scales with that coefficient, it makes sense that growth rate would, at this component of expenditure. That makes sense. What also makes sense is why, the fact that mice and rats and guinea pigs and sheep are right on this line, as you would expect for a well-behaved mammal. And then down below you see the reptiles. That's also sensible because they're cold-blooded. So for any given body mass, a mammal is going to expend far more resources because they're warm-blooded than a reptile, right? So naturally there's going to be more to devote to growth and other functions, right? And so you have this shift up in growth rate compared to reptiles. So that all makes good sense. What doesn't make sense is the fact that humans are right on the reptile line. We're pretty much spot on with it. And we can what we can do here is look at animals of about our body size, right? So right in here, let's look at the mammals that are right at our body size first, right? So smaller to larger, humans are right here in the middle. There's several orders of magnitude difference in our growth rate compared to comparably sized mammals, right? Very noticeable. And then when you compare us to the same do the same thing with the reptiles, we're pretty much right in there with pythons. All right, so there's a mystery here. Humans grow at the pace of a reptile during childhood. Why is this? Now, one commonly cited idea within anthropology through the years is the idea that the brain is unusually costly and it requires slow growth in order to conserve resources. Right? So if it's costly enough, you have to kind of subsidize brain development by slowing and reducing expenditure on other functions, including growth. So many have written about this through the years, and it's kind of an intuitive idea, but it's never really been tested. And that's really what we're gonna dig into today with this analysis. Okay, I'm going to talk about some of these traditional explanations for slow childhood growth. Then I'll tell you about our study, which quantified energetic costs of the brain and tried to put this idea to the test. And then we'll have some ideas at the end that kind of implications for anthropology, for life history, evolution, and other things. All right, so let's start with some background. So this is the pattern of brain growth in humans and in chimps, and you can see that we just continue to grow. Our brains end up much larger than our closest relative. Primates as a whole are very encephalized, but humans are pretty much off the charts compared to most other primates. Right, so we're way off here. This is log again. If it wasn't, you would see that this is quite a bit off that line. Now this all matters in part because brain tissue is very costly energetically. It's about 14 times the metabolic rate or the metabolic expenditure of muscle. Right? It's very costly tissue. And all of you are sitting here right now at rest as adults. Your bodies are probably going through about 100 watts on average. Right? So you're heating up the room at that rate, like a 100-watt incandescent light bulb. 20 of those watts, or about 20%, is your brain. Now 20 watts doesn't seem all that bright for a brain, right? It seems a little dim, but it's not, because if you look at most other mammals, it's in the order of 3 to 5%. 3%, 4%, that's pretty typical of that energetic pie is going to the brain in these other species. You see here spider monkeys and chimpanzees higher, but not as much as the adult human. Now, of course, this is in the adult human, right, where the brain size is actually quite small relative to the body when you compare it to, say, a newborn. And so naturally, that figure is going to be higher early in life. And back when I wrote this review and compiled all these data, the estimate that I could find from the literature had us at about 60% of our brain metabolism, 60% of the body's metabolism going to the brain at birth, right? So given that, if this is even approximately correct, it might suggest that there's something to this idea, that the brain is costly enough that it's going to require trade-offs in other functions, right? And this might go some way towards explaining our slow growth. Now, if that's true, we can come up with some more targeted hypotheses to test that. If our costly brains explain our slow growth, then the ages of slowest body growth should also be the ages of highest brain energy needs. Right? It shouldn't just be that we grow slow as a species. There are periods when the brain is particularly costly. We should see the trade-offs there. We should see slow growth at those ages and not at others. Now, how do we go about evaluating this? We've already got this metric of the percentage of RMR going to the brain. This is a good way to think about it because this is kind of saying, of the overall pie of energy that's available to the organism, what size slice is required by the brain? Which tells you how much it's kind of dominating the body's expenditure and how much there's going to be the strength of the trade-offs with other functions. Now the problem with this is that until our paper at least, there wasn't a whole lot known about the energetic cost of the brain and how that varied developmentally. And the approach that had been taken, the one kind of estimate that allowed some estimation of this across development, started by taking the metabolic rate of the brain, and this was measured in adults, and then multiplying that through. So this is per unit gram of brain, and you multiply it through by the size of the brain at different ages, and then you divide by the metabolic rate of the body, right? And if you do that, you're going to expect it to be relatively high early, and then gradually decline, right, as a kind of continuous function, and sort of converge on that 20% point that I talked about earlier, right? And in fact, when Holliday did this back in the 80s, I plotted the data, but these are the data that he produced, and that basically conformed to that expectation. And this particular paper led to this figure that's been cited quite a bit, that at birth, 87% of resting metabolic rate goes to the brain. Not the 60% that I talked about earlier. From this method, this is what he came up with. And I was always bothered by this because it just seems implausible. I think, I don't remember the exact RMR of a baby, but it's around 100, might be 115 calories. So we're talking something like 13, 15 calories, something like that, to run the heart, to run the liver, to run everything else in the body for a whole day. It just seems implausible. And if this was correct, then I think this idea that there's a trade-off between the brain and growth, we could pretty much discount that because there's no evidence from these data that that exists. Okay, so here we have growth rate. So this is weight velocity, so the rate of weight gain. It's initially fast, it drops down to a low point in childhood, and this is the pubertal growth spurt, right? So the age when body growth is slowest, the childhood years, 4, 5, etc., this is an age of high brain needs but declining brain needs. There's no obvious connection between these two, right, if that's in fact what the curve looks like. The problem, though, is that the per gram metabolic rate of the brain is not stable with age. That was the assumption that allowed them to create this curve. They took the expenditure of a gram of adult brain tissue, measured in an adult, and just multiplied it through, under the assumption that it would be stable. But we know that's not the case. And studies that showed this is a classic study by Harry Chugani from 1987. He was a co-author on the paper that I'm going to be talking about, the data that I'll be talking about. And it was actually these data from this paper that were really instrumental to the analyses that we ended up doing, because it's really the only data of their sort for humans. So what this involves, you give somebody a radio-labeled glucose, right, and then they consume it, and then you put them in a scanner, and you can measure the rate of uptake of that glucose, right, because it's radio-labeled, and so you get a little heat map that allows you to quantify within different regions. And you can also do this globally, or you can do it within different regions of the brain and so on. And when Harry did this is what he found. So this is cortical tissue, right, so here's some adult controls. So this is sort of the adult level. And you can see that at birth, it's quite a bit below the adult level of expenditure, but it rapidly rises up to more than twice the expenditure of adult tissue by mid-childhood, and then it declines. And so what's going on here? Well, first of all, let's look at this. Let's look at the actual PET data so you can kind of see what this would look like. You can see that the brain is pretty dim early in life, and then it gradually becomes more bright. It seems like it's kind of peaking out in this age range here, and then it declines down to the adult level. So what's this all about? Why is each gram of brain in this age range using so much more energy and much more glucose than, say, the adult brain or the newborn brain? Well, it all comes down to synapses, because synapses require a lot of energy to maintain their gradients of potassium and sodium. Right? So you've got ATP that's just driving this kind of constant pumping action, and the more synapses that you have, the more energetic cost there is. And this shows you the time course of synapse formation and pruning that happens in the human brain, roughly. Okay, so you've got a period of synaptogenesis early on, right? And so things like, anybody who's had a newborn, you know that they can't really see or hear particularly well when they're first born. And so that's some of the first stuff that comes online. You have this proliferation of synapses related to seeing and hearing, right? And then later you have language proliferation, and then you have higher functions like executive function that are sort of stretched out into childhood. And what's going on here, the reason that you have an increase and a decrease is that you have an initial proliferation of redundancy, right? So you have lots of extra synapses that are formed, and as you use your arms and speak and look and use all of these different aspects of your body and your biology and your behavior, you're firing certain pathways, and those pathways that you fire, you retain. They're kept. And the ones that never get fired basically are pruned away, right? So it's a process of synaptogenesis followed by pruning. And there's this period, this sort of temporary period of high energetic requirements that underlies that. So here's some of the very few human data that actually show synaptic densities. Classic data from Huttenlocher from the 70s. And you see this increase in synaptic density related to synaptogenesis and then pruning, big data gap, but then getting down to the adult level, okay, later in life. And so when we look at Chugani's data, this is what's going on. You're getting this increase. You're starting off with actually fewer synapses early in life because that process hasn't begun yet, right? So that's why each gram of brain in the newborn is actually lower than the cost in the adult, but it rises up during that period of peak synaptogenesis, and then it declines with pruning. So what this means is, if we go back to this figure, that it clearly is going to need some revising, right? each gram of brain at birth is less costly than in the adult. So this is an overestimate. This is going to have to come down. But by mid-childhood, it's about twice the cost of that gram of brain in the adult. So that's going to have to go up. And so we hypothesized that the curve might look something like that. And in fact, I drew this little ugly curve long before we had the data. So that's what we hypothesized. And so we'll dig into this now for the rest of the talk. Look at these data, see what they actually tell us. What are the costs of the human brain, including the large hidden energetic expenditure related to synaptogenesis, stuff that's revealed by the PET data? And then when we factor these costs in, do we have evidence for this trade-off between growth rate and the cost of the brain as hypothesized? Here, this is actually a culmination of an NSF hominid grant that we had. It was a 5-year grant that had different components, a lot of people involved. The piece that I led was this piece. Others led other pieces, but these are the collaborators on this particular paper. All right, so the method. So we had Chugani's data, which I've already shown you some of, and we looked at average glucose uptake rates within the cerebrum, the cerebellum, and brainstem by age. And here you can see fitting a curve through one of those curves, one of those sets of data. And then, so that's going to give you how many grams of glucose is a gram of tissue, of brain tissue, using per unit time. So then what you need to know is the size of that particular structure and how it changes with age. So for that, we use volumetric data from MRI studies. And there was mostly a longitudinal study that we used, and we filled in with other sources. And so the idea is you've got this per gram rate of glucose uptake, you multiply that by brain volume, and then you get the total energetic cost of the brain. And when you crunch all that, this is what it looks like. All right, so this is glucose use in absolute grams per day by the brain in males and females. Everything's higher in males because their brains are larger and their bodies are larger. There's no, when you account for body size, there's no difference. And a couple things to point out here. Notice that the curves are entering at 15 years of age. That's because that was the last non-adult PET data that Chugani had. We decided not to try to extrapolate it out. Okay, so we just ended there. And a couple things to point out here. So things are peaking out at about 5 years of age in absolute terms. And at that point, the 5-year-old's brain is using about twice the glucose in absolute terms as the adult brain, which is remarkable, especially when you take into account that the 5-year-old's body is 1/3 the size of the adult's body. Right? So if you really want to think about how demanding this is on the body, we have to account for body size. And so the next graph, what we do is that ratio. What percentage of RMR is going to the brain? When you look at it that way, this is what it looks like. So it's peaking out, again, 4 to 5 years of age in both males and females, at which point the brain is accounting for about 2/3 of the body's resting metabolism, right? Which is pretty remarkable. It's really dominating things. This dotted line below is daily energy requirements. So it's doing the exact same calculations, but not based upon resting metabolism, based upon total expenditure. So that also includes like physical activity and growth and other things. Okay? So either way you cut it, there's a big peak in childhood. And so that leads to, we can start to answer our question then. Are the ages of high relative glucose use also ages of slow weight gain and vice versa? And I'm happy to say the answer is yes. So let's take a look at this. First of all, we have cross-sectional data. And so the way that we're estimating growth rate or growth velocity is just calculating a first derivative of this. And when you calculate that first derivative, it gives you the velocity. So it's kind of a cross-sectional estimate of a weight velocity. That's what it looks like. And doing this in SD scores, so this is z-scores or standard deviation scores, so it's unitless, so we can compare it to other things. And when we superimpose upon that the glucose curve that I've already shown you, there's clearly some correspondence there. The period when the brain is at its peak expenditure, when it's really dominating the body's energy budget as a percentage of RMR, is the age at which growth is at its slowest, right? So it seems like there's something going on there, but we can take it one step further. And instead of plotting these both relative to age, we can plot them relative to one another and see if there's a trade-off. So as the brain becomes more costly, do you see growth rate going down and vice versa? And when you plot it, this is what it looks like. And when I plotted this was the prettiest thing I've ever plotted in my whole career. I really was shocked at how cool this came out, because if this was a perfect inverse linear relationship, it would be that dotted line right there. and it's pretty much right on it. So let me walk you through what's going on. So this is at birth, and clearly there's something else going on here. We can talk about that, but it really starts about, it's sometime in the first year of life. We can go between 1 and 2 years of age. At that time, the brain is becoming more costly, and this is because of two things. Between 1 and 2 years of age, the brain is growing in absolute size, right? So brain growth is still happening, and you have that synaptogenesis. So you got the increase in synaptic density per gram of tissue. Both of those things mean that there's a big increase in the percentage of RMR going to the brain during that window. And during that period, weight velocity is decreasing. And because these are both in SD scores, notice that there's about a 1 unit increase here corresponding to about a 1 unit decrease there, right? So the changes are inverse and proportionate. And it's that proportionality that accounts for that linearity, right? That nice inverse linear relationship. When you get down here, okay, this is the point between 4 and 5 years of age when the brain is at its most costly. Okay, it's farthest to the right. It's really dominating the energy budget. And that's also the age at which growth rate is slowest. So that's where the peak corresponds, the peak in the brain costs corresponds with the trough in weight growth. And then over here on the other side, let's say between 7 and 8 years of age, it's going in reverse now, right? So now you have brain growth is done, but what you have is synaptic pruning. So you have that pruning in the brain is actually becoming less costly per unit tissue, right? And so this is moving back to the left, and growth rate is speeding up. And so that's what's going on. And it looks like, I think we've got good evidence for this trade-off in these data. That's males. Here's the female data. It's very similar, except they're going into puberty earlier. So this, whatever's happening here is happening earlier there. And when you plot it relative to DER, daily energy requirements, it still, I think, conforms pretty nicely to that idea of a trade-off. Okay, so let's have a few points to wrap up then. All right, so we started with this question of, we have a pretty good read on why we wean early, but why do we grow so slowly during this period? And I think our data really provide pretty good support for this idea that there's an energetic trade-off that underlies this. The ages of peak RMR of the brain are also the ages of slowest body growth, and when you plot them relative to one another, you see pretty good evidence, I think, for a linear inverse relationship, starting in early infancy and ending at about puberty. So what all this means, I think, is that our findings suggest that the slow pace of human childhood growth reflects a trade-off with the unusually high metabolic needs of human brain development. I think that's the kind of fundamental punchline of all this. But there's other things that come out of this, right? So the idea isn't so much that slowing of growth accounts for the entire extra costs of our brains. Those are not necessarily going to be balanced. If anything, the brain clearly is requiring more energy than is ever devoted to growth. And so it's very likely that there are going to be trade-offs with other functions at this age. And that's a hypothesis that comes out of this. We can expect trade-offs perhaps with immune function or physical activity. Other kinds of expenditure should also be kind of in a trough at this age. So I think we've got some good fundamental insights here into why we grow so slow, but also why humans are kind of unusual in having this really pronounced pubertal growth spurt. I mean, a lot of species have a growth spurt at puberty, but ours is particularly pronounced. And I think you see that we're deferring the costs of growth. Like we're slowing our growth so much that we kind of have to compensate by having an increased pubertal growth spurt later. So I think we get some insights from our data into that feature of our life history as well. One quick little thought about paleoanthropology and what this might mean for that subfield. Of course, the fossil record is often used to try to infer brain size, which is a basis for trying to infer energetic costs of the brain. And I think that our findings really point to the limitations of that sort of assumption, right? We see that there are, you can look at the capacity, the cranial capacity, you can calculate that from the fossil record. But that doesn't necessarily tell you how costly the brain is, because like in our data, the cost of each gram of brain varies 2.5-fold just in a 5-year period of development. And if we continued this on, it would actually go down and substantially, right? It would continue to change in later development. And so this is entirely hidden. You can have cranial capacity, but you have no idea what's actually going on with those dynamics of per gram energetics that are hidden from view. And so speculatively, might this give us some insights into Neanderthal life history? I mean, here, by some accounts, first of all, we know that they have relatively similar brain size to modern humans. And by some accounts, there's evidence that they had a faster growth rate based on dental data and so forth. So really expensive brains, presumably, if you just look at the brain size, and yet they're managing to mature earlier. And so might it be that their brains actually are a little bit energetically less costly than ours, and might that account for their ability to grow faster than us, right? So that's one idea. And I'm going to end with one final point. It's one that I'll develop a little bit. And that is, who pays for the extra costs of human brain development, and how do we buffer brain metabolism? And one point to keep in mind here, before we get into this, is that brains are not only costly, but they have a constant metabolic need. So if you are under severe nutritional stress, you can shut down a lot of your functions. Physical activity, for instance, a lot of organs can slow down. You can't really shut down the brain because you have to continue to maintain those action potentials, synapses, right? And so a big brain is not only an energetically costly brain, but it's a vulnerable brain. And so let's take a look at this. Let's look at the first point first. Who pays for the cost of human brain development? Well, one idea about this, going back some decades, was Bob Martin, who proposed this idea of maternal metabolism hypothesis. And this is based on not just primates, but mammals generally, and kind of an allometric approach, looking at lots of species and seeing what scales on what and so forth. And based upon that work, he hypothesized that brain expansion in mammals has been limited by maternal provisioning capacity in utero. So it's really the size of the newborn's brain that's kind of setting the trajectory. And the size of the newborn's brain is limited by kind of a ceiling of what the mother can invest during pregnancy. So that was his idea. And Steve Lee has looked at that idea and other sorts of ideas, looking kind of variation across different primate systems and so forth. What I think is interesting about our data is it shows pretty clearly that, yes, of course, the mother is bearing a large cost of the human brain, for sure. But this big peak is clearly occurring after the age of complete weaning. So when the brain is really peaking out in terms of its cost and its vulnerability, we're cut off from maternal metabolism. It's not, maternal metabolism is out of the picture. So that's the first point. Let's move to the second point of how do we buffer brain metabolism? It's a vulnerable organ. And one obvious candidate there would be body fat, right? So if you have a big vulnerable brain, you better have a big battery to keep it going. And our data give us some interesting insights into that, right? So here we see that when the brain is very costly, we see that weight gain is very slow. And we can look at this not just in terms of weight gain. I ran some figures to try to estimate fat deposition. Okay? And when you do that, this is what it looks like. This is in males. You can see that fat deposition is very high early in life, but by the age that the brain is peaking out, it's basically negligible at that point. So almost no fat is being put down at that point when the brain is really costly. And so this is what it looks like in female. It's very similar, except it starts rising earlier with earlier puberty. So this leads us to a bit of a paradox. So this is showing body fatness is measured by skin fold thicknesses in a human population, and the age when brain needs are at their peak is the age when body fat stores are basically bottoming out. And the paradox here, you can think of this as an analogy, a mortgage. If you have a mortgage on your house, you have to pay that every month. It's a couple $1,000, $5,000 here in LA. I have no idea. High, right? And if you lose your job, you still have to pay it, or you're going to have to foreclose on it, right? So it's not negotiable, and that's sort of what the brain is. And so the bigger your mortgage, the bigger your savings account better be to buffer those periods when you lose your job, right? So what accounts for this? How can we go out on this limb and have this massive mortgage at a point when our savings account is basically bottomed out? It's an odd situation. And what I think it points to is the importance of social buffering, that this becomes very important by this age, and was likely a prerequisite to human encephalization. People have written about this, many. And so we got two components of that, one of which I've already alluded to, which is allocare and provisioning by kin and non-kin. And then the other is cooperation and food sharing. And the people in this room know way more about this stuff than I do. So this is not going to be news. But if we look at a typical mammal, okay, the mother is the sole support network, right? The sole source of calories through breast milk. And then a typical mammal With weaning, you're basically going from breast milk to self-provisioning. There's no intermediate kind of baby food stage like we have, this long period of provisioning. Now in us, of course, fathers are often part of the picture, so that's interesting. There are other species in which that's the case, and it's not uniform across all cultures, but fathers tend to be a part of that system. But of course, it's actually a much bigger cast of characters that's part of our system. And it's highly flexible. It depends on what society you look in, what household you look in. Different folks are playing different roles, right? And what's interesting about this and what's important about this is not only do you have many different sources of food and care, but these individuals themselves are embedded within broader networks of cooperation and food sharing, right? So this is a very effective buffering system. And we can think about this very simplistically and schematically with this figure here. If you have 4 individuals who are not sharing and they're going off on a hunt every day by themselves, this person gets a kill, hoards it, the other people go hungry. You know, day 2, this person happens to get a kill, these people go hungry, and so on, right? So it's kind of a feast-famine scenario. Now, if the same situation you have food sharing, this person gets a kill, keeps most of it, but shares a lot of it. There's reciprocity on day 2, 3, and so forth. Because on any given day, not everybody comes back empty-handed. That means that everybody tends to eat on every day, right? In this system of food sharing and cooperation. And Bruce Winterhalter, this is some analyses that he did a long time ago showing this. So this is showing the coefficient of variation on foraging returns in relation to the number of individuals that you share with. And so the more people you share with, the variation goes down, right? It's exactly what I was just showing. And this is what you see in human foraging populations. So here, he, we, and Ache, Gervin, and others have written about lots of families are receiving those calories. And here's a study among the Hadza, economic game study in which honey sticks were provided, and then they were doing an economic game, and they were just kind of following the flow of these honey sticks. And you can see it's going to genetic kin, to in-laws, to friends, small, big, and high donations. The findings of the study aren't that important. What's important, I think, is you just see that there's a social metabolism, that things flow through society. Calories flow through our societies in a very redundant and effective way. And I think it's this social metabolism, right, which really comes on board at this age, helps us explain how we manage to go out on this limb and have this massive mortgage at an age when our savings account is so low. You don't necessarily need a huge savings account if you can borrow from wealthy friends, basically. And I think that's basically what we're doing. So these brain data, the brain energetic data, I think they give us some insights into some fundamental features of the life history. I think they help explain why we grow as slowly as we do. They tell us something about why our puberty is as pronounced as it is. But I also think it gives us some insights into the nature of human cooperation and its importance to the overall package that is the human life history. With that, take some questions. Yeah.
So you mentioned adiposity, and without having access to the kinds of longitudinal or cross-sectional information that you have for humans, is it possible to compare across species such that you can explain the sort of remarkable fatness of human babies compared to other primates, for example? That is,
yeah.
Do their just looking at their growth curves by themselves, does that give you enough leverage if you extrapolate what the expected brain demands are going to be?
So that opens up a whole big story, and it's a good question. The first paper I wrote was all about that. It was all from 1998 or whatever it was. Basically dug into this question of baby fat and showed that we are unusually fat as a species. We have much more fat. I mean, the data are sparse, but based upon the data that are out there, we are 3 to 4 times fatter than you would expect for our body size, right? And so I proposed that there was a link with the brain. The basic story that had been out there, people recognized that we were fat. But the story that had been out there was sort of a naked ape scenario, right? It was about hairlessness and thermal regulation. But when you look in the literature, there wasn't a lot of evidence for that. I mean, it was hard to find actual data to suggest that that's what was going on. And so I proposed that brain size might be, a reasonable alternative explanation. And we have such large brains, and there's so much nutritional turmoil early in life that you've got to have a bigger battery to keep it going and so forth. So I still think that could be true, but when you look at our data, it might, at first, if you just look at it on the surface, it would seem to negate that idea because we're peaking out in brain energetics at the age when body fat is at its lowest. So it actually goes counter to what I was proposing for the newborn period. But the more I've thought about this, I actually don't think it's contradictory. I think that they can both exist, right? And the reason is that the sources of nutrition, I'm not answering your question, but I want to answer this first because then we'll get to the cross-species question second. I think if you look at the sources of nutritional stress, it changes a lot. Okay, so early in life, the main sources of nutritional stress are infectious diseases related to weaning. And infections have as their symptoms anorexia, so kids stop eating, and diarrhea, vomiting, things of this sort, right? And all of these, what they're doing is they're basically cutting off kids from caregivers. They're cutting off kids from outside energy sources and forcing them to rely upon internal sources. So if you stop eating, you have diarrhea, you're not digesting your nutrients and so on, you better have onboard reserves, to deal with that. And infection really peaks out very early, but as kids get sick, their immune competence increases, and so the burden of infection is minimal by childhood in comparison to early in life. And so I think by 5 years of age, we have these massively costly brains, but because the source of nutritional stress at that age is going to be ecological or social, it's not going to be infection, then the buffering actually comes into play and works at that point. Okay, so that's the first answer to the question. I think that fat plays a really important role in buffering the newborn's brain because of the nature of the nutritional stress. But by childhood, it's a luxury that can't be afforded in a sense, and you have to divert those resources into the brain and rely upon the social network. Okay, can we, is there even anywhere near enough data to start to tease apart whether or not that's actually, phylogenetically supported? I've been able to find data on maybe like three species, I think, of non-human primates in newborns, and it's it's a real limitation. I mean, it's the kind of thing that I'd love to find a graduate student that wants to run with and generate those data. But in theory, it's doable. With the data that exists, I don't think it is. Really long-winded question, but yeah, or answer. Sorry, yeah.
For that pathogen explanation.
I'm sorry. For
that pathogen-related explanation for infant baby fat, how is that human specific? I mean, how does that explain the extra adiposity of human infants versus, because wouldn't the same concerns over pathogen exposure be true for weaning and other species?
Yes, but their brain size is, so it gets back to this idea that the brain is like a mortgage, right? It's like this, it's a non-negotiable expenditure. And based upon our data and other people's data, the brain around weaning is probably consuming close to 60% of resting metabolism, whereas it might be 10% in another species, right? So that's the idea. It's the combination of unavoidable nutritional stress because the immune system is not up and running, and so you're necessarily vulnerable as a result of that. You're going to get sick. It's just part of the deal. It's how you wire, it's how you get immunocompetence. That's how you build your body, your suite of antibodies and so forth. But it's a combination of that with this kind of this non-negotiable, very high expenditure. So that's the idea. But again, it'd be wonderful to have enough data to actually test that across species. But yeah.
So I'm wondering, when you have malnutrition at different points in childhood, and you've got this trade-off, sort of cognitive versus physical growth kind of trade-off going on, if then you would expect that if malnutrition were to strike at particular points in childhood, if you would be more likely at some points than others to see sort of cognitive deficits as opposed to deficits in stature or something like that. And if you could predict when those time points would be and then go out and actually try and
Yeah, no, it's a really good question. If you look at the literature on nutritional interventions and the effects of nutrition on brain and cognitive development, brain growth, all of that literature, so this is like the international nutrition type literature, right? Basically all of that literature, or almost all of it, focuses on the period of rapid brain growth. The assumption is that when the brain is growing rapidly, this is going to be the critical period. We've got to buffer brain growth. I think our data suggest that there should be another critical period. And it's going to be kind of invisible because there's not a lot of brain growth happening, but there's very costly brain development happening. And so, yeah, I think that there's a prediction that nutritional stress, at the 4 to 5 year range in that area should have long-term cognitive effects. As far as effects on stature, that gets complicated because there's really different effects of nutrition on final stature in the first couple years versus later. So what normally happens, like if you're nutritionally stressed at say 4 or 5 years of age, you're going to mature later and you're going to end up the same size. Okay, so the main effect of malnutrition on final stature is there by 2 to 3 years of age. And it's partly because of the hormonal changes that are occurring and how growth is regulated and so on. But that's considered sort of the critical period. So I wouldn't necessarily expect to see an effect on stature, but maybe you'd see it on delayed maturity or something. Yeah, might be one way to look at that. Yeah.
So isn't the first year of life the period of the highest growth velocity and also the period where a substantial amount of brain pruning is occurring? So how does the nutritional intake account for both of that? And
Brain pruning.
Unrelated question, are there any studies of the caloric content of mother's milk in other primates compared to the human? Do we have much more fat or sugars in mother's milk compared to other primates?
Yeah, well, with respect to the first question, I wouldn't say the first year of life is a period of brain pruning. It's a period of synaptogenesis. It's the opposite. So I think that the net, if you go back to those curves that I showed and the synaptic density curves and so on, you see that synaptic densities are increasing during the first year. And so what's happening in the first year, yes, growth rate is high, but it's rapidly decelerating. So the growth rate is going down rapidly while the brain is becoming rapidly more costly. So I think, the data support that idea. The first half year of life is odd, though. It's that part, it's the little hook that before you get into the period where there's that clear trade-off, there's that little piece, and I don't actually know what's going on there. I haven't been able to figure that out. But the second question, remind me?
About the quality of milk.
Yeah, okay. There's a huge literature on that, and I don't know it that well. Most of the work that I've seen there has related milk quality to the kind of style of rearing. So if there's species that are cash, I can't remember what they're called, caching species, where they kind of, like a seal or something that leaves the pup for long periods of time and then they go off and hunt, right? And so in those species, what you see is very energy dense, high fat milk because they're going to be basically fasting after that for a while. We're an on-demand nursing species, and so our milk tends to be lower in fat and higher in carbohydrates. It's like lower quality because it's kind of, the suckling's happening across the day, kind of throughout the day. That's the work that I've seen, is it's kind of related to that aspect of the ecology, not so much to the brain. But it's an interesting question. I mean, you could definitely propose that there might be some interesting things going on with like omega-3 fatty acids, short-chain fatty acids, things that are structurally important for brain development. Yeah. Yeah.
Back to Brooke's question, I mean, when there are big population-wide food shortages, the
Mhm.
Dutch famine or particular droughts or
Yeah.
periods in Ethiopia when everyone was hungry, aren't there any data showing the vulnerability of the 4 to 5-year-old age period to that?
I don't know of any, but no one's looking at it because most of the stuff that's been done, like in the Dutch famine study, for instance, has been sort of testing the prenatal environment, right? Or maybe the early infancy environment. But they don't look at 4 to 5 years. There's not been a good biological model to test there. But I think that these data should hopefully inspire people to do that. I mean, I think I would hypothesize that, if you're exposed, I mean, it's hard to say because you also do have a lot of growth going on. Brain growth is also very vulnerable. So maybe, 5-year-olds compared to 7-year-olds or something would be a test when brain growth has basically ceased to try to isolate this effect of this energetic piece that's independent of growth. But yeah, I don't know of anybody that's done it, and I'd like somebody to try. Yeah. Yeah.
Pure speculation,
Mhm.
and I'm pretty sure you're going to say no one's looked, but it occurs to me that it would be a remarkable adaptation if we came equipped to detect these kinds of societal levels of poor resources and malnutrition and so on, and delay brain growth in a way comparable to the delay of somatic growth.
Yeah.
That we have a surge during puberty. Do you know if there's any evidence of this kind of facultative shift where you do it a little later
Yeah.
if you have to?
You know, I don't know of any. Like, somebody who studies child development might have behavioral data that would speak to that, and I just don't know what that would look like or what the data are. This is actually one of the next questions that we hope to be able to get at. You know, this is like this kind of cross-sectional snapshot of a, quote, typical human or something, right? It's pretty crude. The next step, you know, PET is very invasive. The reason that we have these data is they were actually collected here. This was at UCLA. Harry Chugani was here. He ran a PET facility, and kids get referred to him because they might have, you know, lesions in the brain. And when he would scan them and look for the lesion, if they didn't have one, he'd put them in this normal data set. So that's where the data come from. You can't ethically just go out and recruit kids and subject them to all this stuff, right? So, what's that?
Or you can't starve them experimentally.
No, but yeah, but my point is that we don't even know how variable this is across individuals. In like early, you know, fast matures, slow matures, etc. There's no information on that, and so we're spinning up some methods that would use MRI that are not invasive and don't involve any sort of injections or anything. That, you know, where we could at least start to look at individual variation. And the first step towards getting at your question would just, you know, if you've got somebody who's a fast mature somatically, do you see any differences? You know, are things accelerated in terms of brain development compared to somebody who's a delayed mature? We don't even know that. That would be a first step. And then, you know, yes, look in the context of, you know, nutritional stress and so forth. I think it's an interesting idea. I mean, at a gut level, my you know, you got to wonder like how canalized the developmental process is, right? Like, body growth is pretty uncanalized. It's pretty fluid and flexible. And like menarcheal age has gone from 17 to 12 years, you know, 100 years, you know, in Europe. So there's a lot of inherent plasticity in that, and it seems like it's part of the system. Brain development might be, I don't know. I mean, it could be flexible or it could be kind of rigid and set. But I think it's a good question. Yeah.
Were you surprised that it was 4 or 5 and not more like 7, 8, 9? Did it really not surprise you? I mean,
Oh, you mean?
there's still some synaptic creation going on past 4 and 5.
Yeah.
Some relevance, perhaps there's another blip of creation just before puberty. But I guess I was surprised just how early this was. And is it just that there's so much subcortically going on at those ages?
Yeah. See, I don't know that the developmental cyclet and so on to the same extent, right? So I wasn't coming at it from that starting point, so I wasn't surprised. But I'd like to talk to you if you think that it should be later, or if there's a lot of stuff going on that is. Yeah. Go ahead.
Actually,
If it's on that top, because.
it's not exactly.
Okay, because I had one more point, and it'll come to me. Go ahead.
How do you suppose the brain pays for all the brain organization that happens at puberty? So according to your data, there's a lot of weight gain, which makes a lot of sense, of course, when that happens in puberty. But at the same time, there's also a lot of brain development, not volumetrically, not in brain growth, but certainly in ways that I think would tap into the metabolic resources.
Yeah, and I don't know. I mean, if you look at the synaptic density data, it's not like there's another peak. No, there's very limited data, unfortunately. There's a pretty big gap in a lot of the data at that age. Does that wiring, I know my point was going to be, and this will be related. It's not that synaptogenesis stops or starts, it's the relative balance of synaptic creation of new synapses and pruning that's constantly happening across the life cycle, even in adulthood, right? So it's the balance between the two that leads to these kind of gradual shifts towards greater synaptic density in a way. But synaptogenesis is, I think, happening in me right now, you know, even though I'm not learning much. But what's that? Yeah, exactly. But there's enough pruning that it's just stable. So it's just the kind of balance between the two. I don't know of data that show like a secondary blip in synaptic densities or PET-related, increase in per gram uptake. I mean, maybe it's something very limited to particular, fairly local brain regions. So it's not like a global, really costly function at a global level. It might be one thing. I really don't know. I would love to talk to you, though, more about this because I don't know that literature. And, how this stuff sort of interfaces with that, I think is a really interesting question. Yeah.
So, and maybe this is what was motivating Andrew's question, I don't know, but if I understand your conclusions, then middle childhood in humans is just the inevitable cost of the earlier expenses and the deferment, if you will, time that it takes to catch up to a growth rate that might otherwise be the expected primate rate. And I'm wondering what your thoughts are in comparing that with various adaptive stories that people have spun about the importance of middle
Learning.
childhood in human culture acquisition and building human capital and all the rest of it. I mean, it's certainly the case that if that is an inevitable cost, then you might as well do something with that period,
Yeah.
right? That is, selection can operate to optimize behavior given that delay in maturation. But that's a different account of what middle childhood is and
Yeah.
why it's there, then it's just the cost that we can't get away from.
Yeah, it's a great question. And I think that, if you think about what is the benefit of having really costly brain wiring processes, it had to go hand in hand with learning, right? So, I assume there were more complex forms of culture and so on, information acquisition. That's the benefit of it, right? And so I don't really necessarily see this as like an alternative to a learning-based idea that we need the time to learn. I see it as all kind of coevolutionary. Whatever, through serendipity or through non-serendipity, still need to think through this, the energetic costs of learning create the time that one needs to learn, right? It actually leads to a slowing down and a stretching out. And I think our data would suggest that energetic trade-offs may have been primary in the sense that it was kind of the thing that drove the whole process. But clearly there was an iterative, constantly, iterative process in which the benefits of having more complex brain development went hand in hand with learning, and it all stretched out, right? I don't have a good answer to it, but I don't necessarily see this energetic argument as, say, challenging the idea that this is really a period about learning. I think learning is costly, and that's part of what we're showing here. Yeah.
I'm wondering if maybe I could tie these two things together a little bit. No, I don't know the biology of this at all, but if the synaptogenesis is the really costly part, right, and then the pruning is less costly, but I'm wondering if during the pruning period is when you're actually more likely to see the changes in cognition, so that you've got the energetically costly synaptogenesis before leading up to middle childhood, but the results of that, like the things that we as anthropologists and psychologists measure, are things that you would actually see the fruition of during the pruning period, which happens during middle childhood. So in that sense, I think it could all still.
That's cool. I like that. Yeah, that might be a tidy way to pull it together. So basically, the big cognitive leaps are occurring as you whittle down and commit to something, as opposed to having this big, unformed, costly. Yeah, I don't know enough about the brain to know if that's viable, but I like it. Yeah. Yeah.
Related to the previous question, do we know, you thought to increase energy requirements from the part of the brain, but this has not to do with the size of the brain. So can we correlate this increased requirements with the type of functions that they are maturing at that time? What does the brain do between 4 and 5 years of age?
Yeah.
What type of functions are they, what kind of abilities do they appear at the time?
Developmental psychologists? Anybody? I don't. Yeah, this is exactly the stuff that I'd like to learn more about. It's not my area, I got to be honest. My interest in this came from energetics and growth and so on, so this is the piece that I need to learn more about. And I've asked a lot of people that exact question, like first of all, when you look at that peak, right, that's occurring at that age, it's not like there's one thing happening, obviously. There's many different individual curves. Like I showed early on, there was that synaptogenesis curve where you see the hearing and sight comes on early, and then language comes on, and so on. That big curve is a kind of composite of a bunch of overlapping curves, some of which, overlap to varying degrees and have different time courses. And so that just suggests that all of that stuff collectively is peaking out at that age, but I don't really know, what's going on the down at that point, what's on the rise, what accounts for it, and trying to sort of decompose it into its individual skills. I don't know. And I haven't been able to get a clear answer on it. I've asked a lot of people, but yeah. Yeah, Hannah.
So the story you've told is very much about energy and glucose, but obviously it's about matter, not just energy, if you're talking about metabolism and biochemistry. So, I mean, this is not what you did, but are there ways in which, instead of looking at energy use, you can actually look at changes in the use of different materials? I mean, synaptogenesis and pruning, they don't happen by themselves. They're enzymatic processes that need substrates and all of those things. So is there a potential by which, if you looked at varying levels of material uptake rather than just the energy component of it, you might see something that helps your story?
Yeah. You would need the equivalent of PET data on those materials. And I don't know if, yeah, I mean, I agree. Like, it would be great to know what's going on with structural fatty acids or, amino acids that are part of enzymes and so on. I mean, that's definitely part of the story. What we have available is what is out there, which is basically glucose. And yeah, it'd be interesting. I think overall, if you think about, the sort of life history, evolution literature, I mean, a lot of it focuses on energy for a reason. Like, it really is kind of limiting. It is the big limiting factor, right? And these other things are more trace. They're more like, yeah, I don't know if they would be limiting in the same way. But it's a good, amino acids, I could definitely see as being really interesting. But I don't know of any way to do that. I don't know of any data that would allow you to address it, unfortunately.
Okay.
You good? Okay, thanks.